Hi, I'm Aman. I'm the co-founder and CTO at Mave Health. About just my company, we are a mental health tech company and we are very outcome driven in mental health, and we are doing it through the intersection of neuroscience and the traditional mental health technologies which people earlier used, and we use a lot of things like brain stimulation, neurostimulation to do that. But we are his talk was a great segue into minds, because we are also trying to question a lot about how AI can be used. We experimented a lot on that, eventually decided that no, we will not be using AI. Should be always a human in the loop. And yeah, so that's where we started. The how I structured this talk was, as you guys are like working in AI, working a lot in AI, this is more of a segue into thinking more from a neuroscience perspective, is how you can think of AI and neuroscience, and how what can you learn from the brain, because all we are doing in AI is trying to mimic how brain works, and most of the things in AI are based on that. So, like, the first chapter of the talk is our brain, which is the OG neural network, the first neural network we saw. And I have done a very bad job at making the slide, so I just have pointers. And we start with neurons. And like, you have 86 billion neurons in your brain, and those are constantly chatting to each other to do, even like I'm speaking right now, you're listening to me, you are thinking, you are like, a lot of parallel things are happening at the same time when I'm speaking, and all these are happening due to neurons constantly firing and talking to each other. And if like we go back to the basics of AI, if you make AI model from scratch, you will start with a node which was called perceptron back then, and that was the entire model was very based on how your neurons work, like how your neurons has a axon, a body, a dendrite, and how I'll go in details into that. And like, this is how a neuron looks like. You have like a top body which is like a hairy structure which is called axon. There is this cell body which is called soma, and these are called dendrites. And like, these are like interconnected to each other. So another neuron comes in a similar, you might have seen this picture a lot in your textbooks. And if you see how neural networks look like, the very similar structure is seen here also. So going back to little bit history of neuroscience, which like seems very interesting to me, because we are dealing with mental health, and you might have heard about, I'm pretty sure, antidepressants, right? Medicines which we use to treat depression. And a reason why neuroscience has been an emerging field in treating all these mental health disorders, and a lot of people ask us that if this is so good, why people have not heard about it, and why are you guys like kind of the first in India to do this? And an answer to this is how brains were kind of scanned earlier. So in 1990s, what happened was they used a dead brain to understand anything about the brain, because there was no fMRI machine, there was no EEG machines. So what they did was they cut a brain and saw what a depressed dead brain looks like, and what a normal live human brain looks like. And because it was a dead human brain, there was no electrical signals in it. The neurons were actually not talking to each other, and what they end up seeing there were like deposit of chemicals like dopamine hai, serotonin hai, and they concluded that okay, this is what leads to depression, or this is what leads to a depressed brain, or this is what the difference is. And that's where the whole study of this antidepressant started, where they concluded, okay, neurotransmitters or the chemical messenger actually lead to depression, the deficiency or increase in that. But then like the emerging field of brain computer interfaces and neuroscience grew, and that's when we started understanding neurons. And if you compare like every part of this is analogous to how your AI models work. So for example, you can consider this as an input to your AI model. This is where the computation is happening, and then this is where the synapses are. So any two neurons when like this particular area of a neuron, this is called synapse, and this is, you must be working with weights in your AI models, how this particular layer correspond to the next layer, what amount of information will be transferred to the next, and that's analogous to how your synapses work. To and yeah, a very dramatic picture, but that's how a communication between two neurons keep happening. And a very simple analogy to understand is how you can understand as one neuron as a speaker, a microphone, and another. Sorry, one neuron as a microphone, another as a speaker. When you speak to, and if you are speaking to like quietly, another neuron might not hear it, and that's where in AI you are using you must be dealing with activation functions like ReLU, sigmoid, all these functions where you say keep only when your inputs pass through this activation function, then only the input propagates to the next level. And that's how neurons work also, that they have a, you again this is a very textbook thing, but yeah, like you have a resting potential of your neuron, and whenever your neurons fire more than this threshold potential, then only the activation happens and the data gets transferred to your next state. So yeah, so this was a basic very comparison between how AI and neuroscience are related, and most of the early models of AI or neural networks started from understanding how brain works. Yeah. So the chapter 2 is where we like a lot of research is going on that how can biology help us in making better AI models or better efficient AI models. So that's the second chapter is where we get neural inspiration, basically building AI from biology. So last week there is this very interesting place in Bangalore called Bangalore Science Gallery. Has anyone visited it?
Yes.
Yeah, so I was just there last week, and there was this place where these very small organisms with slime molds. Yeah, so what they did was on the backside of this is a very like not related to neuroscience, but a very good example of how biological computers work. So if you see on the background, there is a map of Bangalore, and on the top what they did was they spread across the slimy molds, and they kept food at almost every important junction of Bangalore. Wherever there is more traffic, like the amount of food corresponds to how much traffic, how much congestion is at every level, and they just left the slimy mold to do its job. And the path so what slimy mold does, it automatically tries to find path across it, and the path it found was very close to the Bangalore map we currently had, and where it was not close, it found a very optimum like path where, so people were trying to find whether the metro structures we are building or the trains we are building, the railways we are building, are they optimum or not, and this found a very good so that's where like a lot of inspiration is comes from biology, where organisms or brain is used to see what's the efficient approach. I've already talked about efficiency, so you know that most of the large language models are trained with like gigawatts of power. Like a big problem with training models is power consumption, and but based on researches, our human brain just consume 20 watts of power. That's like a 1,000-time reduction, and the amount of parallel computation and the amount of things which go in our brain is just like phenomenal compared to LLM model. Definitely LLMs excel, or any AI model excels in specialization, but our human brain also excels in generalization. For example, if there is a CNN which can recognize some picture, that whether the general digit recognition, that was it written 1, 2, 3, you can, but even if it's small, quality deterioration leads to wrong results. But your human eyes are very like brilliant in understanding. If I see Paras in a darker environment, I know how like my brain can recognize him. So yeah, and a lot of things which we see in AI, which is like prediction machines, which are your LLMs, the attention mechanisms, which are the basics of all the transformer models, neuroplasticity, where like a lot of reinforcement learning comes, where you are like training your model, feeding it back the weights, the rewards to make it better. All are somewhere inspired, and like your brain follows a very similar system. So for example, we do neurostimulation, which means that we are, so this is not our device, this is what we have imported from Europe, but we are building a similar device. What it does is you place these are called, so these are electrodes, these are conductive silicon, and you use cellulose sponges dipped in saline water. Right now it's not dipped, but you place the sponges like this, and then you wear this headset over your brain, like over your head. And what it does, it delivers a 2 milliampere of current over your brain for 30 minutes. And what it essentially does, it tries to restore your neuronal activity back to normal. So on the earlier slides when I shown you this action potential graph, so what happens in depression or anxiety, either this threshold potential goes up or most of your neurons do not get to fire, they do not reach the threshold potential. So what neuromodulation or any kind of, like, this is called, this is based on a technology called TDCS, transcranial direct current stimulation, and most of these neuromodulation techniques wants to restore these neuronal activity back to normal. And like I earlier said, because the earlier studies happened on a dead brain, where they understood that in the end it is the neurotransmitter which needs to be increased, but they did not understood why even your brain or a neuron releases neurotransmitters. Yeah, so like most of the principles you see in AI are based on, so this is neuroplasticity, which this is not like a something which you will wear and you will see a difference in your first time. This builds over time. So over like 30 sessions, you will see changes in your brain because your neurons are adjusting itself, like how naturally your brain works. So again, like neuroplasticity is another topic where it teaches us a lot about like reinforcement learning, etc. Hebbian learning says that neurons which fire together wire together, which means that, like, if there are bunch of neurons which are able to fire together based on, so for example, I just heard a beep from a bike. If that initiated some neuronal activity in some few neurons and that led to some other, still I'm able to focus here, not getting distracted by that, because that particular stimuli is not like lighting up as many neurons as much this is doing. So that's where a lot of Hebbian learning comes in. Chapter 3 I wanted to talk because we, like, a lot of developers are here who must have heard about microservices. Like we talked this about, there is a lot of fight going on whether microservice is good or not. But so essentially what happens is, yeah, so if you are diving deep there, so if this is your brain and this is your prefrontal cortex, it's like the largest part of your brain, 40% of your brain is here. And just to like do a sideline is, like, there are two very important parts which generally correlates to depression and anxiety, which is your prefrontal cortex, which is called PFC, DLPFC or PFC, and there is your amygdala. And what happens is during depression, and generally what happens is the activity of PFC is inversely proportional to the activity of amygdala. And what happens is if your amygdala is firing a lot, that's what happened during anxiety. That's what called amygdala hijack, where you are having too many thoughts, and DLPFC or PFC tries to regulate that. It tries to inhibit that. So that's where, like, so if your anxiety increases, then PFC tries to increase its activity so that the amygdala decreases. So generally in a normal brain, if you are not dealing with depression, if you are getting anxiety attacks or you're overthinking, then your amygdala, your PFC kicks in and say, okay, please, like, calm down. Like, we'll just think of, like, I'm giving a very general example, but that's what, like, that's what mean by an inverse proportion, that if this increases, PFC tries to decrease it, and if, like, your PFC is very depressed, then amygdala kicks in and tries to elevate your mood. But too much in any of these direction, like too less in this direction or too much in this direction, leads to depression or anxiety. Now coming back to your question, how neuromodulation works is when you apply a current. So what happens is, like, for our example, we place electrodes here on our forehead, and this essentially what it does, it builds an electric field here. So it's not like the current gets passed to your brain, it's an electric field which gets made, and what happens is your neuronal orientation matter a lot. So for example, if in your brain one neuron is like this, one neuron is like this, and one neuron is like this, this because like there is no electricity transfer between two neurons. It's what's get transferred are ions, like sodium and potassium ions, which get transferred between two things, and because those are ions, like electrical ions, we call them that electricity गया एक neuron से दूसरे neuron में. But actually what is happening is, so like definitely then if there is lot of sodium or potassium ions here, that means this side is mostly positively charged and this is negatively charged. So that's where the electric field part comes in. When you apply a neurostimulation like TDCS, it tries to align these neurons. So if like there is a electric field here, I'm not sure if anyone of you are from electronics background, for like a capacitor, if you apply a positive charge and a negative charge here, like this will try to align itself in this direction. If this is negative and this is positive, there are more sodium ions here, negative ions here, and this what leads to an like a better action potential over time. And if you zoom, like for example, this dendrite is connected to the axon here, and if you zoom into this, what is happening is in between there are transfer of uh, neurotransmitters happening. What happens in depression and anxiety, because like, and there are, so when you say about action potential, it's not just that the entire how we generally consider with AI models कि ठीक है एक input का दूसरे input पे, and there is a sigmoid function, if it passes it will pass through. There are a lot of smaller action potential which get generated across the cell body, and like if all of those I'm coming back to your answer now, like what happens in a depressed brain is because again when the action potential is not, like the neuron is not able to reach the action potential, but what you are trying to target is kind of all the neurons in your head, and most of those neurons which are like talking to amygdala or hippocampus are like sitting very deep inside your brain. So like last month there was a research going on CIP Rachi, and there they got like they are doing neurostimulation on people in coma, so where like there is almost no neuronal activity, and they have seen results like people started blinking their eyes. They are still not attributed completely it was it due to brain stimulation or other treatments they are getting, but there has been some like good outcomes where neurostimulation is like dormant neurons are also getting activated due to electrical stimulation. Right now the technology this is based on is called TDCS, transcranial direct current stimulation. So the DC part comes from a constant current which is applied. So there is a ramp up over 10 seconds, it goes from 0 to 2 milliampere, then it stays as a constant current for 29.5 minutes, and then it ramp downs in the next 30 seconds. So this is how this works. But there are like different classes of neurostimulation device. This one is called TACS, transcranial alternate current stimulation, where you have a like a sinusoidal wave which is going through your brain. So then there are definitely like a much more nuanced protocols, where there is called TPS, where like pulsed current stimulation, where you like rather than having a constant current, you like send kind of random noise to your brain. TACS has a lot of efficacy in, so there is a company called Somni. They use TACS for insomnia, basically treating insomnia, because TACS has more proven results in very like short-term effects. So if you want to sleep right now, you use TACS, and like I'm betting you, like if you use TACS, you will feel sleepy just after the session. But TDCS is more for long-term effects, where you want to treat depression, anxiety, where you want to like have the effects for a longer period of time so that you do not relapse into depression. But again, like it's a very heavily clinically researched area right now. So most of these protocols, like for example, 2 milliampere 30 minutes is right now what we are using. Based on the clinical research happening later, we might come across that maybe you needed like some kind of staircase pattern in your protocol. So yeah, like this is what clinically proved right now, that this is what efficacy is there, but we are also experimenting on a lot of waveforms on how you can use it based on the positioning. So like I said, these are called montages in terms of brain stimulation devices, or more these come more from a EEG terminology, that there are 1010 and 1020 systems where there are like very specific dots on your brain which are like named as FP1, FP2, and like you choose basically if you want to target prefrontal cortex and depression. There is a website called tdcs.com. If you open it, there is a link called montages. So if you open that, you will see a lot of different. So for example, if you want to target period pain, there is a different montage for that. If you want to target so like for different mental health disorder or any other bodily disorder, you can have a different positioning of the brain. So for example, this particular device has 3 electrodes. So you can like choose any 2 at a time. So you can like choose these 2, you can choose these 2, you can choose these 2. So based on where your positive and negative electrodes, you get a different effect on a different. So for example, if you there is a like very highly research going on. So you must have heard about Parkinson's. Yes. So Parkinson's generally like inhibit your motor activity. So this is your motor cortex. So if a lot of stimulation happens in this area, a lot of research is happening in like TDCS or neurostimulation in this area leads to reduction in Parkinson's. So yeah, like different shape of the electrode, what the current intensity, what the current waveform is, what the positioning of the electrode is, everything leads to a different effect in your. So if you see a clinical TDCS device, you will see a very helmet-like structure where there are like different holes where you can just take an electrode, keep it at like whatever permutation combination you want. What we are focusing on more of a consumer device where like you as a consumer, because these are at home TDCS device. So if I give you a TDCS device at home, and at home if you by mistake do a different like combination what was prescribed to you, that might lead to a different effect. So that's where like consumer TDCS devices are generally like very simple, where whatever it targets, that's what this is for. If you want to target a different area, you get a different set of device. But if you go to hospitals, the TDCS devices there will have that big cap where you can like choose where to keep it, but they are also very costly too. So right now, how I consider it as like I was talking to Paras about this also, is we consider right now the devices we have as right devices. The next, like you must have heard about brain-computer interfaces, BCIs or EEG. Those are, I call them read devices. So where we want to reach in our version 2 are a read-write device, where you have the read functionality and the write functionality. Like you said, like we want to close the loops where like, and like you said, like right now we have this and this kind of a protocol. But what if I need a 2.1 milliampere of current, you just need 1.8 milliampere, he needs 1.9 milliampere of current, but we just don't know it yet. And that's where we want to unlock this potential of reading EEG waves from that, getting biomarkers from there, and understanding like how does TDCS is affecting your brain waves, and personalizing your like mental health stimulation according to that. Because honestly, every person is very different, every person's brain is very different, but still we are applying the same stimulation to everyone, because that's what been proven in clinical researches. But that might not be true. So that's where the complete loop comes in, when you apply a BCI device and a neurostimulation device and plug them together. So that completes a feedback loop. That's where the Hebbian principle comes in, like the neurons which fire together is like, it's not just like two neurons talking to each other. I removed one slide, but there was a slide, I was in a different talk where I was talking about, it was a very different audience where they were not developers, so I changed a lot of slides according to that. But there I was showing like you must have seen dominoes falling, right? Like if one domino falls, you see a whole cascade of dominoes fall. That's a very analogy of how neurons also work. Like it's not like just one neuron talk and like it stopped there. If a good enough action potential was like occurs, most of your like neurons will start talking and it will like do whatever action was intended. And like there it was that what if you remove one domino from it, right? You just remove it, or you make one of the domino in between very tall and very bulky. What will happen even if all the dominoes fall, either because there is a dominoes removed, it will miss and all the like rest of the neurons or the dominoes will not fire, or because the action potential in between was so large that it could not trigger that particular neuron, and your activity like stopped there. So that's what this was an analogy to explain how depressed brain works, that in terms of dominoes. Okay, so that was a quick segue into a lot of things. Again, like I think we have discussed more interesting things on what I intended to. Again, I was just trying to understand. So I'll just jump to the final thing, which is error handling. So you must have heard about microservice architecture, where we see that we are not dealing with a monolith structure. There are small systems which should happen and like work in parallel, should be resilient, should be like error proof, etc. And there is a very interesting thing happen in our brain which is called corollary discharge. Okay, what happens is, so for example, if someone else tickles you, you can you can like you start laughing, you get irritated by that. But if you try to tickle yourself, you can't, right? Again, another example of this is, for example, I am like constantly looking here and there. If it was a digital camera, if it was a mechanical camera, what would happen if like the frame rate at which we see the world, you will see a shaky image, like a one image overlapping over each other. But that's not what happens. We see a very stable image. Like even if I blink my eyes very quickly, like it does not happen that you almost see a stable image of the world. And that's where like corollary discharge, the concept of corollary discharge comes in. Is what happens is your brain, whenever some action happens, it was very interesting in terms of hallucinations which happen in LLMs. Is whenever you perform an action, what your neuron does is it tries to recreate that action, kind of makes a copy of that action, sends back the payload kind of thing to your brain, tries to replay that thing in your brain and see if it was a self-induced action or not. If it was, then don't react to it. And so that's where like you tickle yourself and it does not does anything, and it has like that's what helps in like stabilizing your vision in your retina. So that's where the corollary discharge. I'm not able to like go deep into it, but a previous point where LLMs are more like analogous to, it's what I'm trying to speak to all of you guys. I'm not thinking of the next word before speaking. It's like coming naturally. I'm just talking. Like when I'm talking, I'm not able to think. It's a feedback. It's confusing when I think about it. But yeah, but right, like so that's what LLMs does also, right? Like all the previous input lead to the next token. So all this prediction which keeps happening is a very like a general function of your brain, which where you are just doing the next possible thing automatically. It's not just about speaking, it's about hearing, it's about and like another example like he said about schizophrenia. So a lot of times what happens is when I'm speaking, my brain is very easily able to differentiate between my speech and your speech, right? And that's where like my speech, kind of what your brain does, it kinds of like it decreases its amplitude, so you do not get irritated by it. So a lot of like basic example is the insects which call its mating partner using like cricket noises don't get irritated or don't get excited by their own noises, because their brain kind of inhibits their own sound. What happens in schizophrenia is this corollary discharge is kind of disturbed, and your own voice kind of sounds like someone else's voice, and that like disturbs your complete auditory nerves. So yeah, like if something bad happens in any of this, it leads to a mental health problem. Again, like I was just trying to show how like there are different parts, and like this is where the vision things happen, the occipital lobe. So like there are like very parallel things in your brain where like different activities are happening. So this was just an attempt to show that. Again, like I think I've previously talked about this, that like your brain just consumes 20 watts of power to operate, and it's is like inspiration, and like I'll talk to about further chips which are being developed. And what neurons generally does, it follows sparse coding. So definitely like you have 86 billion of neurons, so even 1% of neurons is a lot, but generally only 1 to 2% of neurons are working at a time. Like if you're in a very like maybe we are in a talk, maybe more neurons are firing at right now, but generally only 1% of neurons are firing at a time. And that leads to a lot of new development in AI area, where you want to do sparse coding there also, so that your AI models, so like all the small language models or sparse language models which come, you see like Meta ka Olama was like 128B, say they brought it down to 8B, which is 8 billion. So that's what they are trying to, like, decreasing the number of neurons so that you get a similar enough like output, but in a more efficient structure. Because the next topic is called neuromorphic hardware. So a lot of the, like said, Intel, IBM are developing these new kind of chips, which they call neuromorphic chips, where they are very closely seen. So this is, for example, Intel has this Lohi 2, and this is IBM's True North chip which they are developing, and these work in a very similar way how our brains work. So most of the things which we heard earlier to this was more of an analogy. So like there are neuromorphic hardwares which are being built where, like, rather than going with a always on kind of system, they are going more towards a event driven system, with like how our brain works, that our brain does not like always constantly computing something. Whenever some external stimuli happens, we respond to that. So that's where these people are building neuromorphic hardware. They are like, right now the purpose is to build edge AI, where like on wearables. So for example, if you have an Apple smartwatch, and so if you have a, like a, I will not call names, but if you have a cheaper smartwatch, if you start running, it will not automatically detect that whether you ran, whether you walked, whether it was a bicycle, what you did. You have to manually go there and start that activity, and then it starts tracking. But most of these, like, expensive smartwatches like Apple watches are trying to build this intelligence in the watch itself, that if you start running, it would automatically tries to guess that whether this particular hand movement, does it correspond to running, does it correspond to bicycling, does it correspond to driving. So based on that, it's trying to like automatically like track your things. So that's where like a lot of edge AI is coming in, where like for example, smart glasses are coming in. There are a lot of companies which are building this for blind people. So there a lot of computation needs to happen on the hardware itself, rather than like your data goes out to the cloud, then it comes back, like it's too late for a blind person to walk till then. So a lot of these neuromorphic hardwares are being used in edge based like wearables on like AI on the edge, because also they are energy efficient, because like these only fire when something happens, not every time it's working. And like another segue into this is like SNNs. Yeah, so spiking neural networks. These are again a new like field of neural network, like RNNs, CNNs. Now there are SNNs, where this is based on like neuromorphic hardwares used kind of SNNs inside it, where rather than depending on what the input was, you kind of just depend on when the spike arrived in your system. So it's like the temporal dynamics basically tells that it depends on time a lot rather than the value of the inputs. So again, like energy efficiency is where like all of these hardwares are aiming towards. Yeah, I mean, so more and more I was reading on neuroscience and AI, I realized that we have a very symbiotic relationship there. Like neuroscience is trying to drive AI and AI is trying to drive neuroscience. So a lot of labs are trying to produce virtual laboratories where, like, rather, because like the only way you can experiment on a real brain is when you get a dead brain. Real life experiments do not happen a lot. So a lot of people are making virtual laboratories where you can simulate your brain in that labs, and you can run like a lot of simulations. You can understand maybe what's happening inside a brain. So like things like DBS or deep brain stimulation, or what Neuralink is doing, they are trying to reach the neuronal layer. How do they work? Sorry, I'm digressing. So they kind of try to measure. So rather than, so see, most of the devices like EEG devices, or there's another technology called FNIRS, which is near infrared. So they use light, like blood level things. Again, so most of these, like the basic of all these reading devices is reading electrical signals back. Whether you get it through accumulated voltage over an area, or if you can directly measure like what's being communicated between two neurons. Again, like I'm not an expert in what happens inside your brain when like Neuralink electrodes comes. Yeah. Well, the purpose of Neuralink, so if you, there is a very interesting podcast on the Lex Friedman channel where the entire Neuralink team comes in. I'll definitely advise you to take a look at it, where they are like, if we want to go more of a telepathy layer, where we, whatever you are thinking, so a lot of people who have used Neuralink, they say that, not of people, one people who has used Neuralink, he says that he could take an action before he actually, like his thought actually reached his mind. It's a very difficult thing to comprehend, but like he was able to move the cursor before he actually decided in his head, okay, I have to move the cursor. Like the EMG waves actually like decided that his hand should do it before the brain did it. You must have seen the Meta glasses which came, right? They have a wristband along the side with it, through which you can control the device. So those work on EMG. And like a very interesting thought there also is that if you try to move your like finger like this, you won't see any EMG signals. But if you move it like this, you will see EMG signals. So yeah, like in the end, it's like the signals which are driving it, not like, it's very difficult to comprehend a lot of things which happen. Yeah, so the entire ECG, EMG, EEG, like how you talked about closing the loop, a lot of people are working on like combining all of this. So OpenBCI is a company in US which does a lot of all of these interesting things. They have a TED talk where they flew a drone in a room full of people just using brain computer interface. And there you can see that they combined EMG, ECG, EEG, all these three like sensory devices. Yeah. So they combine all of this to fly that drone. So in the end, like the future looks like where you have all these read devices combining and giving data, and finally you are